Hello and welcome to SciLux, the podcast where we talk about scientific developments and research in Luxembourg. Please don't forget that we are powered by Research Luxembourg and straight from the studios of RTL City. Our guest today is Dr. Johannes Meiser, Head of Cancer Metabolism Group in the Department of Cancer Research at LIH, so that is Luxembourg Institute of Health. Johannes studied biology at the Saarland University, and this is where he also got his PhD at the Institute for Molecular Plant Physiology. He first came to Luxembourg to work at LCSB, then moved to the Beatson Institute in Glasgow. Now, as an FNR Attract Fellow, he leads the Meiser Lab, which recently celebrated 4 years of existence. Johannes, thank you very much for coming today.
Hello, thanks very much for the invitation.
First of all, congratulations on your celebration, because, I am always interested when you tweet something that is not about science, but more about celebrations. And I've noticed that at your lab, you have this tradition of doing something special altogether, which was nice to see.
Yeah, thanks very much. It's always nice to reflect once in a year, at least, on the things that happened during the last year. And it's astonishing that we are 4 years already on.
And it's also, I know, something that is very dear to you and important, leading a team, right? It's not only research. We also recently, during the FNR Awards, were discussing this whole idea of mentorship and actually helping people to get somewhere with their research. And I noticed that you are really paying a lot of attention to that kind of leadership side of it.
Yeah, that's true. It's actually also, I can say, maybe one of my passions, and maybe also because I'm still young and know that it's good and important having good mentors that support you. And it's also a great fun, I have to say, to build the team and see the individual strength and make sure they work together and help them.
But today we meet not only to discuss that, I'm sure we'll be mentioning some of your colleagues too, but more about, as usual with me, definitions and understanding, getting to the core of your research. And of course, what can I start with? You're the head of Cancer Metabolism Group, so tell me what cancer metabolism is.
Yeah, so cancer metabolism in general describes one of the disciplines within cancer research. So specifically, we are focusing on, as the name says, the metabolism, meaning that how cancer cells use different nutrients that are available in the tumor and in the circulation to proliferate and also to metastasize. And as also the name says, although we have a general interest in metabolism, we specifically study that in context of cancer cells to see also what might be Achilles heels, as we call it, that can be exploited for therapy.
So it would mean finding a way for not providing the food to the cancer cells, or actually figuring out what type of nutrients are the most important, which side it is. Is it kind of more preventing the nutrients to come, or more kind of understanding what they are? Where are we with this?
Yeah, so you touched actually 3 different disciplines, I would say, with that. All of that is ongoing. I mean, looking back, there was the very early discoveries 100 years ago by Otto Warburg and colleagues. And
that's the Warburg effect.
Basically rooted into the Warburg effect, which is still highly debated today. And then in recent decades, I would say metabolism research in the field of cancer revived. And one of the initial attempts, and still, of course, very relevant, is to target specific proteins, so-called enzymes, that are involved in converting the different nutrients. So to cut off basically nutrition supply to the cells. And out of that, also more recently, concepts developed where people try by nutritional intervention to force, let's say, cancer cells in specific niche, and then maybe combine that together with other therapies to increase the efficacy of these treatments. But this is still very early, I would say, but very exciting at the same time. So there's first companies and also small trials started now. And of course, to me, it appears quite attractive, because if we would indeed be able, just by altering, let's say, the diet to the patients, we could already improve the efficacy of certain treatments, would be quite attractive. But of course, as with any new trial, there's also downsides. And I think it's very important to first study that in more detail to really understand what are the pros and cons. How can we then effectively use it?
Of course, we mentioned this Warburg effect, so I have to ask you, because there are always two options. One option is I will tell our listeners to go to the show notes, because of course there is a lot discussed that we just can't get into detail with. But sometimes I just need to ask you, because I'm pretty sure that not everybody knows about it. So what was it actually? Or what is it?
Yeah, it roots back, as I said, to the work by Otto Warburg. And I have to say, to me, that's extremely fascinating. Back in the days with the, let's say, limited tools available, he could study metabolism of cells already quite successfully. And he observed that cancer cells excrete a lot of lactate, so much more than normal healthy cells. And he hypothesized at the time that cancer cells have a defect in a specific cellular compartment that is called the mitochondria. And therefore, cells need to or can only break down the sugar, the glucose, into lactate and excrete it. We know that from sports, where people that go over their training limits increase their lactate levels. However, as Warburg described it, and this is the key difference, he postulated that these cells are doing that even in the presence of ample oxygen, so that it's not due to an oxygen limitation, which we would refer rather to the Pasteur effect, but to the observation that these cells cannot use their mitochondria, and therefore they run this, as we also call it, aerobic glycolysis, so high glucose rates in presence of oxygen.
Okay, wait a second. So what you're saying is that this lactate is the same when I get muscle sores when I do too much exercise?
Yes, that's exactly the same metabolite, just in that case is when our body and the circulation cannot supply fast enough oxygen that we need for respiration, then the cells can engage on an alternative system to still make some energy without the mitochondria that are central to that. In that case, the glucose is only partially metabolized into lactic acid, which then goes into the blood and can regenerate it via the liver. But we also know that this works only for a very limited time, right? For example, that during a sprint, this works, but it won't work during the long distance run. And cancer cells can engage a similar program where they metabolize excessive amounts of glucose and thereby generate lots of lactate. But I have to say, and here we are coming a little bit to the modern research of cancer metabolism. I mean, first of all, today we understand that the mitochondria are by far not defective in cancer cells. Instead, they are highly functional and very important, not only to generate the energy, but also to contribute with many different functions to oncogenesis in a large sense.
The thing is that in our discussion right now, I just have a feeling we're in this dark forest and you gave me like 10 paths to go and I don't know where to go. But there is one definite path I want to go. You mentioned something about oxygen and I know you prepared the pub quiz question for our listeners related to oxygen. So can you please ask the question, and listeners, remember the answer will only be at the end of the podcast.
Exactly. The question is related to oxygen. We all know that we inhale oxygen and it's essential for our life. But the question is actually, where does the oxygen end and why is it so important to our system? And what is it doing and how is it getting out of our body again?
Thank you very much. Answer, as usual, at the end of the podcast. When I was preparing for this discussion today, I was also very much trying to look at your papers, the ones you publish. And I have to say, I just spectacularly failed to understand even the title. Okay, so the one that I know that you were particularly proud of was Mitochondria Preserve an Autarkic One-Carbon Cycle to Confer Growth-Independent Cancer Cell Mitigation and Metastasis. Is that right? I read it correctly?
Yes.
Okay, so what part of research is that? What are we talking about here?
Yeah, that's basically the continuation of the work I started during my postdoc times at the Beatson in Glasgow, where we discovered a phenomenon that we call formate overflow. But back at the time, we just made this biochemical observation where we observed that cells excrete large amounts of this molecule, formate, maybe comparable to lactate, but just via very different biochemical processes. But then, of course, we had the question, why are cells doing it? So what could be the selective advantage that this phenotype developed? And with this, basically, we started in Luxembourg as a question in my group also, and we observed early in the beginning when we collaborated also with Simone Niclou, who is working at the LIH as an expert in glioblastoma, and together with her help and different other models where we got access to in Luxembourg, we observed that higher concentrations of this metabolite formate can actually activate the movement of cancer cells. So we call that migration or invasion. So what we observed, and we can model this in the laboratory, is that when we add more formate to the cells, the speed of the movement, let's say, increases. And based on that observation, we aimed to understand what are actually the metabolic processes that support this program. So how is it possible, and what is essential to drive it? Because if we would know what is needed for it, we could maybe also try to target that to inhibit the spreading. And this is an early step during the process of metastasis. So when cancer cells escape from the local tumor site and build the metastasis, which is one of the biggest problems in the clinic also. Then Nicole Kieveler, who is a postdoc in my group, focused specifically on this question, trying to understand what is needed for this movement process. And we used a drug that is very well known in the clinic. It's called methotrexate, or in general terms, you can call it antifolate. It's also used in certain autoimmune diseases. And it's very well known to target this metabolic part that is responsible to make the formate molecule. But then we realized with our biochemical assays that this is actually not true, that only a part of that process is inhibited. And then we found out that this part that is still active is actually quite important to promote this movement process of the cell. And since the drug that is still used today successfully in the clinic can inhibit the proliferation of the cells, so the division, it is not successful to inhibit the movement. So with this work, we observed that basically the movement program and the proliferation program of the cell can be separated and promoted by different metabolic processes. And of course, this is, again, early days, but this is, I would say, a first publication where we are able to point this out. And now we try to better understand also to find the details. But apparently this metabolic pathway that makes formate is quite important for this process.
Okay, a lot of questions. First one, so we understand that the cells excrete formate. But you were saying that you kind of gave them more formate. So why? I mean, why are they excreting it? Or what's the process behind it?
Yes, that's a very important question that we also try to understand. So we give formate from outside because we want to model high concentrations of formate in the space that is around the cell. Of course, we always need to work with simplified models. And in principle, there's two possibilities. So either the formate that accumulates outside of the cell can then dock on the cells or maybe also act on other cells and trigger an effect. Or it is something in the cell, and just because the concentration in the cell is so high that parts of that automatically also flows out, and it's more a signal of very high local concentration. But by giving high formate from the outside in our model system, we can also indirectly increase the intracellular concentration. So at the end, we just use it as an attempt to increase the concentration of this molecule and to see then what happens when the concentration is high compared to cells where the concentration is low.
And do we know what happens when there is a high concentration in normal cells, not cancer cells, of formate?
This we haven't studied so far so extensively. We looked a little bit also in collaborations into immune cells because we know the immune system is very important also in the tumor microenvironment. Of course, the tumor is not only cancer cells, it's a mix of other cells, including immune cells. And we were interested, or we are still interested, to see if formate actually that is excreted by cancer cells can impact the activity of immune cells, either way, if it is positive or negative. But this is still very early days of research, and so far we can't make conclusions on that I would share here.
Of course. It's great you mentioned Professor Niklu, because I talked actually to Dr. Gołębieska at a certain moment from LIH about glioblastoma and their research as well. So if listeners, you're interested, that part of the cancer research and glioblastoma is there in the library of the podcast. But I wanted to ask you exactly, when you decide to study cancer metabolism, do you have a preference, should I use that word, not really, for a certain type of tumor or not? How do you, because, you have to choose something. It's not that you just do everything, right? So which cancer, which tumor, should I say, you prefer in that such study? Is it because of the format that maybe works better, you can observe it better? What's the choice here?
Yeah, in principle, I'd say there's maybe two type of labs. Either there's a specific focus on a specific cancer type, trying to really understand that, or
like glioblastoma in this case, right?
Like glioblastoma in that case, or, and this is maybe more applying to my lab, there is a particular interested in metabolism, and we are interested to find concepts and principles that might also apply to different cancer types, or then also to see, by identifying a specific phenotype, is this generalizable, or is it maybe specific to a certain type? And therefore, we do not focus on one particular cancer type, but we have a few model systems that we use. Of course, here we focus a little bit, because doing everything together is too much. So the two model systems, or the three model systems we use a lot, is glioblastoma on the one hand, because we have also, due to this collaboration with Anna and Simon, a very good interaction and access to very good models to study in particular these movement effects, or the local invasion, let's say. But on the other hand, also we would like to understand in how far this format molecule can contribute to metastasis at the whole systemic level. And glioblastoma is not a known tumor, it's a local problem, a very big problem in the brain, but not perfectly suited to study the whole metastasis. And to look at this part, we focus on breast cancer, where we have also established good models in recent 4 years, and there we can look at the full metastatic cascade, starting from a local tumor in the mammary fat pad up to metastasis in the lungs, for example. And here we also took advantage, I have to say, and I would like to highlight this also, that's why I'm so happy being in Luxembourg, that we have a good collaborative network established, and we work a lot with Elisabeth Letelier from the University of Luxembourg, who has a lot of expertise in this field especially, and she's also an expert in colorectal cancer. And that's the third model we are also studying, which also gives rise to metastasis. So depending a little bit on what we want to look at, we work more with glioblastoma, breast, or colorectal, but these in principle are the three tumor types we are studying.
And when you're talking about cooperation, it just means that somebody created a lot of those cells in the lab and you can just get some of your share, because you said we have a very good model. So does it mean that someone just went and created that model? I mean, the cells themselves, and there are a lot, so you can use them? Or what do you mean by the good model?
Yeah, and I mean, an important part of research in general, I would say, is to be collaborative, and we want interdisciplinary, because we are so specialized with our methodology and knowledge that if we want to study a complex disease like cancer, I think it's extremely important to work with others together. And Elisabeth, also Anna and Simon, have developed during recent years excellent models. This be brain tumor model, for example, or breast cancer model, to build the whole metastatic cascade. This involves animal models, but also models that can be studied in the laboratory in vitro in the dish to replace animal experiments. And there is a lot of expertise available. So we get access to that. Also, for example, patient-derived models that we can use. Xenografts, and that can also be studied again in the dish to work with models that are maybe more relevant to the human disease. And on the other hand, we can then contribute with our expertise in biochemistry, metabolism, analytical chemistry to measure different metabolites, and bringing those two parts together, relevant models and the biochemical expertise, we have a better setup to hopefully reach findings that are meaningful, because that should always be the goal at the end.
And how do you study metastasis? I just started thinking, so you have those cancer cells, And what, you just attach them to healthy cells and wait until they kind of proliferate and cross, or is that it?
Yeah, there's 3 levels, I would say. Purely in vitro based approach, where we can study, for example, the movement of the cell or the local invasion, when we can have an artificial matrix, we call it, that represents also what is seen in a human tissue. And there we can seed cancer cells that we can culture in a dish on, and then we can follow by different means, by microscopy, for example, how these cancer cells are able to degrade the matrix and cross a certain barrier. So by that we can model how in vivo cells could go through a tissue. Then there is another approach where we have a piece of tissue. It really comes from an organism, and then we can inject the cancer cells into that tissue and there monitor again how the cells move through the real tissue structure, which maybe comes a bit closer to the real system, let's say. And then the third attempt is when we really want to see if it impacts the whole metastatic cascade. We also use mouse models, because there we have the whole complex system, a local tissue, a primary tumor, the blood circulation, also the presence of the immune system, depending on the model. And we can see if the cancer cells really survive the travel, let's say, through the system and reach other organs. And that's unfortunately still very hard to model completely in vitro, and therefore it's still very important, I think, for this type of research to use this as well, because otherwise I think it would also be rather risky to directly translate results to clinical trials on humans.
Yes, so that means that you do this parallelly in some kind of a way. You try that one way of studying metastasis, and then you go for the other one, right? Just to kind of check different conditions and different reactions.
Yeah, most of the work we start, or the projects also start, in purely in vitro based systems in a dish. And then when we think we have something that is really interesting, we go up to the more complex models, let's say, to see how relevant the finding really is. Because sometimes it happens that we observe something in the Petri dish, but then it's not recapitulated in a more complex system. And maybe this also relates to a point that I mentioned in the beginning, that today we are also quite careful on how we culture the cancer cells, because traditionally there's standard cell culture media, let's say, that provide the different nutrients, including glucose and so on, of course, where the cancer cells can grow on, and they need the nutrients to survive. But these media are far from physiological conditions as we see them in our human body, in the circulation. I mean, the most simple example is maybe the blood glucose concentration in humans is around 5 millimolar. And in normal cell culture medium, it is around 25 millimolar. So 5 times the concentration. And that was maybe also, or is one of the reasons why Warburg back at the time observed so much lactate production, because it's just this excessive amount of glucose. So what we also use to validate our findings is media that are more closely to what we see in the blood.
And what is the reason that there is so much more glucose in that media? Just because they would grow faster, so you could observe the effects faster?
They grow faster, and it's easier, let's say, to culture the cells, because you need to imagine in our organism, there is a constant supply from the blood flow. And so by having this running system, the concentration of glucose is stable. But when you have the medium in your dish, there is no constant supply. It's just whatever is in there, and it's consumed over time. So this means if you start with 5 millimolar, the concentration quickly drops and then gets below this threshold. So starting with a higher concentration makes it easier to culture the cells. So again, it's whatever model you use, you need to be aware about the positive and negative sides, and then try to do alternative experiments where you have different advantages and disadvantages. By combining then different attempts, we hope, of course, that the findings, become more relevant at the end.
Yes, I understand that. These dishes you mentioned and that kind of a method of growing cells, is that what the metabolomics platform at LIH is for, or you're using it for something else?
The metabolomics platform is an analytical platform. So here we measure our isolated metabolites from the cells. So we culture our cells in the laboratory. And then when we want to measure the metabolism, at the end, we break basically the cells. So we collect them and break them. And this has to go very fast in seconds because the metabolic rates are happening in seconds. And then we isolate the metabolite fraction from the cells. We separate this from proteins, DNA, for example. And then we use the platform to measure them specifically. So it is a chromatography. So that's a column basically where we can load the mix. And based on their chemical properties, they go faster or slower through the column, and thereby they are separated. And then one after the other, they are injected into a second machine, and this is called a mass spectrometer. And with that, we can determine very precisely the mass of the molecule. And each molecule, or most of the molecules, have a unique mass. And by that, we can separate them. And then we can measure at the end, basically according to the cancer cell state, let's say, what are changes at a metabolic level.
So was it also how you found out about the format overflow? Similar method was used?
Yes, exactly. Yeah, it was, looking back, it was it's a simple method, but the challenge was to measure this specific molecule. So one of the standard approaches we do is to collect also the cell culture medium at the beginning and the end of the experiment. As I have just explained, the glucose concentrations can decrease. And when we do this over time, we get, of course, also a measure on how fast do the cells consume a certain nutrient. So, for example, in hypoxic conditions, that can be faster than in normal oxygen
Hypoxic?
Conditions.
What's that?
Hypoxic is, we call it when the oxygen concentration is very low.
Okay.
So this can be, for example, also in the tumor when there's not a good blood supply and therefore less oxygen supply, and therefore hypoxia occurs. And so to measure identify this format phenotype, we collected cell culture medium at the end and at the beginning of the experiment. And then we measure the set of metabolites that is changing in the medium. And there we observed that actually the format concentration increases quite dramatically. But the challenge here was that in normal approaches, as we apply that also on the metabolomics platform, format cannot be measured because it's very small molecule that cannot be measured with this attempt. So we had to apply a specific chemical procedure to make it possible to measure it. And this actually also happened in Glasgow, also together with another colleague who was working there and was working on a related method where he had to develop something. And then we realized this also works for format. And again, having a collaborative approach, we got access to the method, and then I applied this method to our experiments, and then I was able to see actually the cells secrete a lot of formate.
So you couldn't find out about the format using mass spectrometer, that's what you mean?
It's also mass spectrometry, but a different chromatography.
Okay.
Because in the normal approach, format would just go through and would be lost. And we have to first induce a chemical reaction to make the molecule bigger. And then it can be separated, but it's also a different chromatography. So there's liquid and gas chromatography, and we need this gas chromatography in that case.
Okay, very complicated, but congratulations on finding something out. I always wonder, I talked a couple of months ago also to Professor Szymański from LCSB about mass spectrometry and trying to find, the unfindable, if you can call it like that, because for me, her mission is just completely crazy. This whole idea of not knowing what is inside and still trying to, through different measurements, figure out what are the unknown chemicals. It just really sounds like science fiction, but that's what she's doing. So kudos to her for that. But exactly, just kind of understanding this, how do you end up finding that something like that is excreted? Because we mentioned Warburg and the fact that he definitely had less platforms, analytics, possibilities and all that. But I always wondered, how do you start looking for something if you don't know that you will find it? I mean, it's just, so if you can explain this to me, to my simple thinking brain, is it that you just by accident found that format at a certain moment and then started looking for more? Because you said with the method we were using, we couldn't really see it, right? So how do you start thinking, there is something special there? Was it just, I guess what I understand is sometimes it's total accident, right? Was it like that too?
It's a mix of everything, I would say, but I agree that often it's just pure curiosity. Or you do something and have a random idea and test it. In 99% of the cases, it leads into nothing. But sometimes you find something that is really puzzling and then you want to understand and then you dig deeper. Here it was a little bit similar because that was maybe also a lucky coincidence. My supervisor in Glasgow, he was a mathematician and also very strong on computational approaches to model metabolism in silico on the computer. And he played a little bit around with the metabolic network and thought about alternative ways, how cancer cells can make energy basically when classical supply chains, let's say, are not functional anymore, right? And then his model predicted that there is formate excretion, but it was not really clear how this, magic computer box came up to this solution, let's say. And we knew at the time that the best described way where formate is used by cancer cells is to make nucleotides for DNA. And this is, of course, essential for any proliferating cells. But especially in cancer cells, it was assumed that then the demand for formate is even higher, right? Because they need to make so much DNA. So from that point of view, it wouldn't make any sense to excrete this building block. But as then the colleague developed the method and we were wondering what's happening there, I just gave it a shot and collected some medium samples and measured, and then indeed we could observe it. Then it got actually interesting because then we tried to understand, of course, we wanted to know where is it coming from? Is it controlled really by the biochemical network, or is it just maybe from some dying cells or whatever? And we came up with hypothesis to test it. And then we used other drugs to interfere with the metabolism. And we predicted, okay, if we do this, it should become more. If we do that, it should become less. Test like this. And then it's like an empirical approach. And at some point you come to a working model where you can basically say how it works. And that's how we found this formate overflow phenotype. Yeah.
So very interesting story. I would say probably weekend work involved as well. When I was talking.
Other, yes.
Yes, you can't do it within an 8 hour a day situation as well, for sure. And also creative approach, But of course, most of the times it's just nothing. I remember when I was recently talking to Dr. Ibrahim Boussad, he said that, it's just exactly experimental biology in general. It's 1% success. Most of the time it's just frustration. So I think it defines it very well in some way or another. Okay, when I was doing research for our discussion today, I also came up with the discussion of catabolism and anabolism when you talk about cancer metabolism. Can you tell us a little bit more about these terms and why are they important?
Yes, so that's basically the two principles of mammalian metabolism, I would say. So cancer cells, as I said, can or have to consume nutrients, or any cell of our body. And there's two processes. On the one hand, these nutrients are used and needed to make energy to catalyze any given work, also muscle work or anything else, or neuronal work. And on the other hand, as when we want to grow, we need building blocks. Similar as you want to build a house, you need the bricks and everything. So cancer cells, of course, want to proliferate and to grow, and therefore they need proteins, DNA, and lipids for the membranes and so on. And this has to come from somewhere, and it comes from the nutrients, right? So anabolism describes the process of taking nutrients and building them into more complex molecules to proliferate, and catabolism describes the process of breaking down nutrients, or like burning fuel in an engine, let's say, to make energy. And the energy is, of course, also needed to build the more complex molecules. So it's an interplay of both.
And is there any way we can actually influence that? In the sense, because we're trying to stop them from growing, right? So is there something that we can use to, not provide the energy?
Yes, of course, this we try this. And as I mentioned also in the beginning, these antifolates, methotrexate, and also others, they are classical drugs, let's say, to inhibit the anabolic process because they prevent DNA production in cells. And then if they can't duplicate the genome, the cells, they cannot divide and proliferate. Right, but as we have discovered also, this does not prevent the cancer cell to escape and to move, right? So it would not prevent maybe the onset of the metastasis. So there is different inhibitors in trials to target specific routes of this metabolic network to stop the energetic or anabolic program. But the cancer cells are also extremely flexible. So they can, when you take away one nutrient, they often can just switch the metabolism and take up other nutrients to compensate. And on the other hand, they are not only flexible, they are also plastic. And plastic in that sense means they can use one nutrient and use it for very different purposes. So it means when the cell suddenly faces another challenge, they can repurpose the available nutrient for something else. And this makes it, of course, very challenging. So ideally, we find specific situations where cancer cells have a very specific need. And if we understand where this is relevant, then we can maybe better go in with the right treatment. So context dependency is maybe the key word here.
Kind of going back to the precision medicine as such as well, right? And just also analyzing the exact patient, but also the exact cancer and so on and so forth.
Exactly, yes.
I understand. We're going a little bit back and back and back because I just realized I did not ask you one more question that was kind of pretty interesting to me. I thought metabolism is just metabolism, that's it. But I've noticed that in your papers, in your discussions, you have one carbon metabolism, serine metabolism, and I don't know what else. Why are you making these distinctions?
Yeah, that's a certain part of metabolism. And it's right. 30, 40 years ago, probably metabolism was still called a housekeeping function. So it's just running, but it's boring and not needed. And nowadays, it's more and more appreciated also outside of our specific field that metabolism is actually very important to drive this whole process in cancer cells. As I said, there's glucose or other sugars also, but there's of course also many different amino acids that are needed for protein synthesis on the one hand, because it's the building blocks of proteins, but amino acids can also be used for catabolism and anabolism of other systems. And serine is one of these amino acids and the second most consumed amino acid by cancer cells. And the metabolic pathway that is mainly dependent or supported by is the one carbon metabolism. So the one carbon metabolism is mainly supported by the amino acid serine. And it's called one carbon metabolism because it's actually only a part of this amino acid that goes into this pathway. So it's one carbon indeed. And serine has 3 carbons, but only one carbon goes into the cycle. And there it can go different oxidation states and contribute to very different aspects. So it's a very fascinating cycle actually, because it's also split across 2 different compartments of the cell, the mitochondrial part that I mentioned at the beginning, that is also important for energy production, and also the cytoplasmic part of the cell. So that's basically, let's say, the normal part, if you want, of the cell. And in this part, the DNA and all these, the synthesis of the different macromolecules takes place. And as I have indicated, there's many different things depending on this one carbon pathway. And therefore, we dedicate a lot of our attention to this particular part of metabolism.
So you just focus on one carbon that influences everything. It just reminds me, all the discussions I've had so far about, how our body works is just that, okay, we pinpoint one molecule, one substance, whatever. And then actually it's very often some very, very, very small percentage that does something, right? And then it gets diluted. And it's still such an important part of our daily life. It's just mind-boggling, I would say, for sure, at least for me.
Yeah, and I think it's important to realize this, So if you maybe want an analogy on that is if our planet is just a tiny, tiny bit in the whole universe, right? So, and similar, we are studying a tiny aspect of whole cancer biology, but as it is so complex, I think it's also very important to focus on that, but then also to realize, it's in the end maybe one piece of the puzzle that only together with the work of many others hopefully will lead to something, right?
Yes, but we need a lot of, coffee. Do you drink coffee?
A lot, yeah.
Yeah, so coffee break discussions, I guess, right? Because the interdisciplinarity happens also, like, oh yeah, that person has a lab and now let's talk. And then you come up with this solution, right?
Yes.
It's important. I mean, that human aspect and, small talk activities that actually lead to something more, because then you get the ideas for sure. We came back a little bit to the way body works. And unfortunately, I have to already mention that our body clock, or rather the podcast clock, is telling me that we're slowly wrapping up. So we have to go back to the question you asked in the beginning of the podcast. So that's why I'm saying body, because we have oxygen and what happens with it. So can you please remind our listeners what the question was and then tell us what the answer is?
Yes, so the question was, what happens with the oxygen actually that we inhale, and where does it end in the body? And one would maybe think that as we exhale carbon dioxide, which also contains oxygen, that maybe the oxygen would just end in the carbon dioxide that we exhale. But in reality, and then I'm coming back here also to the metabolism of glucose and Warburg, if glucose is not metabolized to lactate but enters the mitochondrial compartment where it can be fully oxidized, it donates electrons to a very complex protein complex, where then the energy, the actual biological energy, let's say, we call it ATP, is produced. And at the end of this long cascade, the electrons together with hydrogen flow together with oxygen and make water, H2O. So the oxygen ends in the water, while the carbon dioxide that we exhale comes from the oxidation of different nutrients like glucose.
So the carbon dioxide discussion, again, perfect. You're just like, our discussion today is just so good for advertising the podcasts that I've had, because we've just discussed some important research happening in Luxembourg related to hydrogen, but also carbon dioxide. So great you mentioned that. But this is very interesting. I had no idea about it. So I was very happy that you prepared that question, because of course the assumption would be exactly what you said, that it does not get where it does. We might also say, okay, why don't we just not provide the nutrients to the cancer cells? Yes, but we will not provide the nutrients to normal cells too, right? And that's exactly the problem.
And I think an important aspect that is also coming more and more clear in recent years is that the immune system plays a very important role in fighting cancer. So it's actually the best natural endogenous system to recognize very early wrong cells and directly kill them. So if we take away our nutrients, we will also weaken our immune system. So it's a very fine balance. And that's also why I mentioned at the beginning, these attempts to try different diets, to support cancer therapy is very attractive. But at the same time, we need to study it in detail to really contribute to the success of the treatment.
Yeah, these are also very popular, discussions in the media, right? Eat this because it's great, right? Or don't eat this because it's cancerogenous or whatever.
Maybe one thing also, as we are sitting here on the radio, is to mention that, and as you say, it's very popular. I have to say, I'm sometimes a little bit concerned when I read on blogs or whatever, where some persons give recommendations when people have cancer, or just like this, what is healthy and improves the combination of certain amino acids. And I would be very careful with this, because I hope I could convince you now that metabolism is very complicated, and also in context of cancer treatment, it's extremely context dependent, and there's not the one solution for everything. And one where we are very clear now, and I just met a colleague in summer at a conference in London again, who presented a lot of data on that, is that antioxidants, where we would think that is very healthy, like vitamin C, is not always helping doing cancer therapy. So there were several clinical trials that had to be stopped because the patients on antioxidants did even worse than the control groups. So yeah, just to say maybe that research is very important to understand mechanisms first before giving recommendations.
Yeah, and not oversimplifying things is also very good. So then we have to finish with this note: go eat whatever you feel like, because then for sure, I mean, the most important thing, you provide nutrients to your body, and we really don't know what's going to happen next, because, well, there is just too many different processes involved and it's way too complicated, but probably not too much coffee, right?
Probably not, yeah.
Yeah, that we kind of agree, or not really. How is it with coffee?
This I don't know, to be honest. I haven't studied it. But what is clear is that a healthy mixed diet and some sport is certainly very beneficial.
Okay, thank you very much for today, Johannes. It was a real pleasure to have you here.
Thank you very much for the invitation again. I also enjoyed it a lot.
And this is it for today. This was Silax, powered by Research Luxembourg, straight from RTL City Studios. And as usual, the usual litany of things. Don't forget to subscribe, to follow us, to rate us, to tell others about us. We are very active on Twitter. We are active on LinkedIn. So just, send us a message if you have any comments, maybe guests you want to suggest. Just remember, they have to be based in Luxembourg, have a link to Luxembourg, do research in Luxembourg. So yeah, we're staying still in Luxembourg, although I'm planning to maybe have some takeovers once in a while, but we'll see how it goes. For now, this is it. Thank you very much. My name is Hanna Siemaszko, and this was Silax.